Hello students, good evening everyone. I hope all are doing well. Anyway, warm welcome all of you to the Infinity Learn JEE YouTube channel by Sri Chaitanya. This is me Surya, your chemistry faculty from Infinity Learn. And I believe I am perfectly audible and visible to each and everyone present over here. If anyone is viewing this live, please give me a hi in the comment box. Okay, finally we are moving to the last part of this particular chapter, that is biomolecules. Here we are dealing with the structure of DNA and RNA. In the previous session we have discussed about the nucleic acids, right? The structure of different nucleic acids. Here we are focusing about the structure of DNA and RNA detailing. After this particular topic, we will be dealing with the most practice questions from the biomolecules chapter. Let's quickly start this topic. And I hope you guys have revised to the topics we have covered in the last classes. If anyone missed the classes, just go and revise the sessions after this class. Okay. Let us start with the structure of DNA. What do you mean by DNA? What is the full form of DNA? It is a nucleic acid, right? So we can call the DNA as the Deoxyribonucleic acid. So we can discuss the structure of DNA into two levels. First one, it is the primary structure, and the second one, it is a secondary structure. Yes, deoxyribonucleic acid, right? Yes, Sabraj, you are correct. So let us start with the primary structure of DNA here. In generally, in the previous class, we have discussed about the polynucleotides, right? How we are forming the nucleotides. There will be a phosphate group, nitrogenous base and the sugar, right? These are combined together to form a nucleotide. So in the case of a DNA or any nucleic acids, right? Any nucleic acids, there will be the formation of nucleotide, or it is known as the polynucleotides. So in the primary structure of DNA, the nucleotides are joined, right? Nucleotides are joined through the phosphodiester bond. Okay, what do you mean by the phosphodiester bond? We have already discussed in the previous classes about the nucleic acids or the nucleotides. Okay, it is a particular bond that is formed between the phosphate group, right? Phosphate group and the sugar molecule, right? Sugar moiety present in the nucleotide is known as the phosphodiester bond. So in general, it should be written in the free 5' end. We will be writing the number of particular carbons. So we can say that there will be the free 5' end on the left and the free 3' end on the right side. Let us see the structure detailedly. So this is our DNA primary structure. As you can see over here, this is our nitrogen base, right? Here it is given as thymine. It is given as thymine, and this is our sugar molecule, right? This is our sugar molecule. In case of DNA, the sugar present is deoxyribose, right? Deoxyribose sugar. Then this is the phosphate group. In total, together, phosphate bond, sugar and the nitrogen base is known as the nucleotide. These nucleotides combine together to form a primary structure of DNA. Clear? Okay, Ankur, what is your doubt? Which portion you want to clear? We have already discussed about the structure of nucleotides, right? This is the primary structure of DNA. Here you can see that this is the sugar molecule. If you missed the class, you can just go and revise the particular session about the nucleotides, right? I have already mentioned that this is our sugar molecule. In the case of DNA molecule, it is deoxyribose, right? So this is the deoxyribose structure, in which a phosphate group, right? Phosphate group is present here, or the phosphoric acid. There is a ester bond is formed between the phosphate group and the carbon, right, present on the sugar molecule, right? So it is forming a phosphodiester bond here. Here it will be phosphoester bond. So these phosphoric acid, sugar and nitrogen base combine together to form the nucleotide. In each case, one of the nucleotides are combined together to form the complete nucleic acid. Here this is the primary structure of DNA. Okay, I hope it is clear for everyone. Now let me move to the secondary structure of DNA, which is given by Watson and Crick, right? James Watson and Francis Crick. So this model is also known as Watson-Crick model. This is the double helical structure of the DNA. This is the double helical structure of DNA. So Watson and Crick proposed the model of secondary structure of DNA. According to this model, you can see over here, right? DNA will be present as a helical structure, right? Helical structure, and it is forming a right-handed double helix. This is our nucleotide chain. What are these? In between these helical structure, there will be some nitrogenous bases, right? There is a particular combination of combining the particular nitrogen bases. Let us see one by one. Then this particular structure or the double helical structure is also known as B form of DNA. B form of DNA, which is the most stable form of the DNA. Clear? Let's see what are the features of the B form of DNA or the double helical structure of the DNA. So, in the case of DNA, there are two strands are present, right? Two strands are present. These two strands of polynucleotides are anti-parallel, which means that they are moving in an opposite direction, right? They are run in the opposite direction. Then, the DNA is composed of the two polynucleotide chains running in the opposite direction, in which they are held by the hydrogen bonding, right? Hydrogen bonding between the nitrogenous base pairs. As we already know, in the case of a nucleotide, there is a sugar molecule, phosphoric acid and the nitrogenous bases, right? So, in the case of DNA double helical structure, there will be the hydrogen bond formed between the different nitrogenous base pairs. So, in general, adenine and guanine, right? Adenine and guanine are the one strand compulsory base pairs with the thymine and cytosine, which means that adenine always pairs with the thymine in the case of DNA, then guanine always pairs with the cytosine and vice versa. Clear? Adenine with the thymine and guanine with the cytosine. Adenine cannot form hydrogen bond with the cytosine, then guanine cannot form hydrogen bond with the thymine also. Is the thing clear? Now the next question arises that how these nitrogen base pairs are combining, how many bonds are formed in between them, right? So if you are taking adenine thymine pair, right? Adenine pairs with the thymine, right? Adenine and thymine. We can say that there will be two hydrogen bonds, right? There will be two hydrogen bonds in between adenine and thymine. So we can represent it as two bonds, right? Adenine pairs with two hydrogen bond in between adenine and thymine, right? Then what about the next one, guanine and cytosine? Guanine combines with the cytosine to form three hydrogen bonds. You will get an idea while teaching about the structure of guanine and cytosine. So here it can be represented as guanine with cytosine having three hydrogen bonds, right? Three hydrogen bonds. Clear? These are the main features of the double helical structure of DNA. If it is clear, just let me know in the comments. Okay, this is the Watson-Crick base pairs. So first one, it is showing which nitrogen base it is. Hi Shankar, good evening. Okay, this is our adenine molecule, right? This is the nitrogen base named as adenine molecule. So it will combine with the thymine, right? So second one, it will be the thymine. Okay, here you can see that there will be sugar molecule also present. So we can call these as the nucleosides, right? Nucleosides in between, in which sugar and base are present. So this is the adenine nitrogen base and this is the thymine nitrogen base, right? So you can see that there is a hydrogen present, right? There is a hydrogen present in the adenine molecule, right? So it will form the hydrogen bond with the next nitrogen base, right? Thymine, which are oxygen and nitrogen, right? So it will form the two hydrogen bonds. Clear? It will form two hydrogen bonds in between the adenine and thymine, which means that there will be two hydrogen atoms are present in the structure of adenine. So it will form the hydrogen bond between the oxygen and nitrogen of the next nitrogen base, that is thymine, in case of DNA, right? Then what about here? This is a cytosine, right? Okay, this is a guanine. This is a cytosine, right? So while observing the structure of nitrogen base here in guanine, you can see that there is 3 hydrogen, right? 1, 2 and 3 hydrogen atoms having the ability to form hydrogen bonds between the other 3 elements or atoms present in the cytosine nitrogen base, right? So it will form 3 hydrogen bonds represented by the 3 bonds here. Clear? Shankar, is there any problem with the online classes? Are you able to understand? Okay, fine. Here we are giving the live classes, right? Every day, 5 to 6, we will be providing the chemistry lectures in this particular channel as the online classes. Okay, there will be some offline centers in Bihar, Patna also, right? For Infinity Learn. Okay, now let us talk about the structure, right? How 2 strands of DNA are forming. You can see over here, this is the first strand, right? First nucleotide chain here. This is the phosphate group. And this is the sugar molecule. Again, this is the base. Here, adenine, right? Adenine combines with the thymine to form 2 hydrogen bond. Then, vice versa, which means thymine also combines with adenine. Guanine with cytosine with 3 bonds. Cytosine with guanine with 3 bonds. So, while observing this structure, you can say that the first nucleotide chain, right? That is complementary to the next one. Clear? Now let us talk about the structure of RNA. RNA means ribonucleic acid, right? So in the case of ribonucleic acid or the RNA, the sugar present is ribose sugar, right? Ribose sugar. In between these 4 nitrogen bases are present: uracil, cytosine, adenine and guanine. So while observing this structure, you can see that in the case of RNA, it is a single helical structure, right? Single stranded structure. Then the difference between DNA and RNA is that the difference in the nitrogen base, right? Difference in the nitrogen base. In the case of DNA, it is thymine. Instead of thymine, in RNA, it will be uracil, right? Clear? This is the main difference between the structure of RNA and DNA. So based on the idea, you can make a table between the differences of DNA and RNA. In the case of DNA, there will be the nucleotide chains, that is ribonucleotides, right? Ribonucleotides are the repeating units. But in the case of, there is a small correction. This is for RNA. This is for DNA. Right? In the case of RNA, ribonucleotides are the repeating units. Then in DNA, it will be deoxyribonucleotides. Then what about the base here? Here the base will be thymine, while in DNA it will be absent, right? Okay, in the case of RNA, instead of thymine, there will be uracil is present, right? Uracil is present. There is some mistakes in the table. So here in RNA, ribonucleotides are the repeating units, DNA, deoxyribonucleotides. Then in terms of nitrogen bases, in RNA, thymine is absent, while DNA, thymine is present, right? Then again uracil, in terms of RNA it is there, in terms of DNA it is not there. In the case of RNA it is single helical structure, but in DNA it will be double helical structure. Now let me move to the application of DNA or the DNA fingerprinting, right? So each and every human have the different fingerprint, right? They have an unique fingerprint. So from the fingerprint we can identify the personality, right? Who is that person? That is the role of DNA fingerprinting here. The sequence of bases, right? Sequence of bases on DNA. That basis on DNA is a unique feature of the person. So based on that, we can use this DNA fingerprinting for identify the criminals. Then identify the dead bodies by comparing their DNA with parents or children. Then identify the racial groups to rewrite the biological evolution. Biological evolution, studying the evolution of new biological species. And the last one, the determination of paternity, right? Determination of paternity of the individual humans and identifying the viruses. These are the main applications of DNA fingerprinting. Clear? Okay, so far we have discussed about the structure of DNA and RNA. I hope it was clear for everyone. Now let me move to some of the practice questions from this particular chapter, that is biomolecules. Here is the first question. If one strand of DNA has a sequence is given here, you have to identify the sequence of complementary strand. Sequence of complementary strand. What will be the answer? The sequence of nitrogen base is given here. You have to identify the complementary strand sequence, which means that we have an idea that adenine always combine with the thymine, right? Adenine always combine with the thymine, then guanine with cytosine and vice versa, right? So we have to write the strand like this. Okay, this is our given strand. Now you have to identify the complementary strand. Okay, what will be the first one here? Adenine will combine with which of the nitrogen base? Adenine with thymine, right? So adenine with thymine, then thymine with adenine, guanine with cytosine, abbreviated as C, cytosine with guanine, then again thymine with adenine, guanine with cytosine, and last one adenine with thymine, right? So this will be our complementary strand given here. You have to check with any option which will be the correct answer. You can see that option D will be the correct answer here, that is TACG AACT, right? So this will be the complementary strand. Yes, correct, Subhash. Then you can give this strand name also here. If it is 3' then it will be 5'. Then the complementary strand will be 5' to 3'. You might be studied in your biology classes in detail about the strands. Okay, here is the next question. This question is based on the peptide bond, right? Peptide bond or the amino acid part. What will be the answer here? A peptide is made up of n number of amino acids, and it must contain how many number of peptide linkages? Recall the structure of amino acids, how these amino acids are combined to form the peptide bond, right? How the peptide bond is forming, how they are forming the peptide linkages, or using the number of amino acids. What will be the answer? Four options are given here, right? Give me any random guesses also. A, B, C or D. Right? This is the peptide bond formation. Hi Jatin Meena. Option C, right? Are you sure, Subhash? Okay, let's take the answer. So, the peptide bond are formed between the two amino acids or the amino acids by the elimination of water molecule, right? So, there will be a bond that is the amide bond. This is known as the peptide bond, right? Peptide bond. So in this particular case, you can see that when we are comparing two amino acids, right? Here we are comparing two amino acids. How many peptide bond is forming here? How many peptide bond is formed here by combining the two amino acids? One peptide linkage or peptide bond, right? One peptide bond is formed between the combination of two amino acids, which means that if you are taking n number of amino acids, right? So the number of peptide linkages formed will be n minus 1, right? N minus 1, which means option D will be the correct answer. So if you are taking 3 amino acids here, that particular peptide linkage or peptide formed is known as a tripeptide because it is made up of 3 amino acids, but it will form the 2 peptide bonds, right? 2 peptide bonds because 3 minus 1 is equal to 2, n minus 1. Clear? Okay, here is the next question, again based on the structure of protein. Which structure of protein remains intact after coagulation of egg white on boiling? What will be the answer? While discussing about the structure of proteins, we have studied about the one property, right? Denaturation of proteins. So here the coagulation of egg white is an example of denaturation of protein. So question here given as the which structure of protein remain intact during the denaturation of protein? Okay, what will be the answer? We have studied about the 4 different structures, right? Primary, secondary, tertiary and quaternary structure. So while discussing about the denaturation of protein, which means if you are changing the temperature or pH or adding any of the reagents or detergents, we can change the structure of proteins, right? We can change the structure of proteins, that will happen to the denaturation, that will lead to the denaturation. So if you are doing that particular denaturation process, secondary and tertiary structure will change, right? But primary structure remain intact. Primary structure remain intact. So here the answer will be option A. Clear? Okay, here is the next question. Naming of the peptide chain. Based on the naming of peptide chain. In order to answer this question, you must have an idea about the structure of different amino acids. What will be the answer? Okay, we have already seen the structure of amino acids, right? In the case of amino acids, there will be two functional groups. One is the carboxylic acid group, another one is an NH2 groups, right? So, if any of the amino acid is having free amine group, right? Free amine group, it is termed as N-terminal, right? N-terminal. Then if it is containing the free carboxylic acid group, it is termed as C-terminal. While writing the peptide name, while writing the nomenclature of peptides, we will be starting from the N-terminal amino acid. N-terminal amino acids, which means that in the case of a peptide chain, that particular amino acid that is having free NH2 group. So here you can see that this is our amino acid, right? This is our peptide chain or the dipeptide. Here two amino acids are forming. You can see that this is the one having the free NH2 group, or it is known as the N-terminal amino acid, right? N-terminal amino acid. Now, so we will be starting the naming with the N-terminal amino acid. N-terminal amino acid. Here it will be the free COOH or carboxylic acid group. So it is termed as the C-terminal, right? C-terminal. So now you have to identify which of the amino acids are combining here. Okay, if you are breaking from here, you can see that these are the two amino acids combining to form these particular peptide. So, I hope you must have an idea about the amino acids. So, I can say that this is the valine, right? This is our valine amino acid. So, this is our glycine, the simplest one, right? Glycine. So, we can start from the naming from right here, right? Right, having the N-terminal. So, we can given the name as. There is one more point you have to be noted, that the N-terminal amino acid will be given the suffix as YL. Instead of INE, it will be given as YL. Glycinyl valine, right? Glycinyl valine. That will be the particular name of this peptide chain. Here it is asking about the abbreviation, right? So we can give it as GLY, right? GLY hyphen VAL stands for glycinyl valine, which means option C will be the correct answer. Is that clear? Okay, here is the next question. Based on the vitamin part, you have to match the column. In the column first, it is given the names of different vitamins. Then in the column 2, it will be given the name of the deficiency diseases. What will be the answer? Four different vitamins are given here. First one is thiamine, then next one riboflavin, pyridoxine and ascorbic acid. Then their deficiency diseases are given here. You have to match the following. Here the chemical names of vitamins are given. So you must have an idea about the chemical name of different vitamins, right? Along with their symbol also, then deficiency diseases. So what will be the thiamine here? Which vitamin it is? Okay, we have already seen that there will be different types of vitamins, right? So the vitamins can be classified into two types depending upon the solubility in water. There are two types: fat soluble. Vitamin A, D, E, K stands for the fat soluble vitamins. Then remaining B and C. This vitamin B is again subdivided into different groups like vitamin B complex starting from B1 to B12, right? B1 to B12. So here the vitamin B1 is known as a thiamine. Thiamine is the chemical name of vitamin B1. Then what will be the deficiency disease for the vitamin B1? Beriberi, right? Beriberi is the deficiency disease for the vitamin B1. So, A to Q. Then what about riboflavin? Riboflavin is the chemical name of vitamin B2, right? Vitamin B2. What will be the deficiency disease? What will be the deficiency disease? Chylosis, right? Chylosis is the deficiency disease for the vitamin B2. So, B for R. Then pyridoxine. Pyridoxine is the vitamin B6, right? Vitamin B6. It have the deficiency disease of convulsions. And the last one, it is ascorbic acid. Ascorbic acid means it is vitamin C, right? Vitamin C. It is having the deficiency disease of scurvy. So, A, Q, B, R, C, P, C, S, and D, P, right? Which means option B will be the correct answer. Yes, option B will be the correct answer. Clear? Okay, repeated questions are asking from this particular topic. You must have an idea about the chemical names of the different vitamins, starting from vitamin A to K, right? Vitamin A, D, E, K, and vitamin B complex. Vitamin B complex along with their deficiency diseases. Hi. Clear? Here is the next question, again based on the peptide linkages. Okay. The question is based on that. A short peptide on complete hydrolysis is produces 3 moles of glycine, 3 moles of glycine, 2 moles of leucine. This is again one of the amino acid, right? Then 2 moles of valine molecule per mole of peptide, right? So, whenever a peptide bond is forming, during the hydrolysis, it will form 3 moles of glycine, 2 moles of leucine, and again 2 moles of valine. What will be the number of peptide linkages in between these peptide linkages, peptide bond? Okay, what will be the answer? Okay, we have already seen that if it is containing n number of amino acids, it will form the n minus 1 peptide linkages, right? So, you have to calculate the total number of amino acids present in this particular peptide, then find out the number of peptide linkages. Okay, what will be the answer? So, it is given that it is forming 3 plus 2 plus 2, 7 amino acids are present, right? 7 amino acids are present here in this particular peptide. So, we can say that there will be 6 peptide linkages. 6 peptide linkages are present in the particular peptide group, right? Option B will be the correct answer. Clear? Okay, here is the next question. The question is based from the topic carbohydrates. Which of the following on hydrolysis give similar monosaccharide units? Similar monosaccharide units. Options are given here. First is sucrose, lactose, maltose and amylose. Okay, we have detailedly studied about the structure of carbohydrates, right? We have seen that there will be monosaccharides, disaccharides, oligosaccharides and polysaccharides. So here is some of the examples of the polysaccharides and oligosaccharides. So you can say that what will be the monomeric units or the monosaccharide units present in these particular polysaccharides. Okay, what will be the answer here? So we have already seen about the structure of sucrose, right? A sucrose is a polysaccharide or the disaccharide that is formed between the alpha D glucose and the beta D fructose, right? Alpha D glucose and the beta D fructose. So we can say that glucose and fructose are the monomeric unit or the monosaccharides present in the case of glucose. Then what about the lactose? So in case of lactose, it is formed between the beta D glucose and the beta D galactose. You must have an idea about the structure, is very important in the examination point of view. Glucose and galactose is combined to form the lactose molecule. Then what about the maltose? Maltose is again containing alpha D glucose, right? Alpha D glucose units. Two alpha D glucose units combines to form the maltose here. Then amylose is the part of water soluble part of starch. Starch is a polysaccharide that contains two parts. One is amylose and another one is the amylopectin. In the case of amylose, there will be alpha D glucose structures, right? There will be a linear chain structure. So it is formed between the two alpha D glucose structure. So we can say that these are the two structures. Maltose and amylose are the two polysaccharides that give similar monosaccharide units. So C and D, which means option D will be the correct answer here. Here is the next question. The positional information of amino acids in a polypeptide chain is called as which of the structures? We have already studied about the different structures of protein, right? Primary, secondary, tertiary and quaternary. Based on that idea, give me the answer. So while discussing about the primary structure, I have already told that in the case of primary structure of protein, there will be the sequence of amino acid, right? There will be the sequence of amino acid. Is there any change in the sequence of amino acid? There will be the different proteins are forming, right? So sequence of amino acids, or in terms we can say that it will be the positional information, right? Positional information of amino acids. It will be given by the primary structure. It will be given by the primary structure of protein. Which means option A will be the correct answer here. Then while discussing about the secondary structure, you must have an idea about the alpha helical structure. Then beta pleated structure, right? Beta pleated structure. Then what about the tertiary structure? In tertiary structure, it will given about the shape of the proteins, right? So it will be two types, globular and fibrous protein. Just recall through the difference between globular and fibrous protein along with their examples. Then quaternary structure, right? Different subunits are present in the case of quaternary structure, for example, hemoglobin. So structure of proteins is also a very important topic in the JEE main examination point of view. Okay, here is the next question, again based on the subtopic vitamins. Identify the correct statement among the following. Different statements are given here. The first statement is vitamin C is water insoluble. Second one, vitamin A is water soluble. Third one, it is vitamin C cannot be stored in our body. The last one, riboflavin is vitamin B6. Which is the correct statement given here? Correct statement. Option C, right? Vitamin C. So we have already seen that the fat soluble and water soluble vitamins. Vitamin A, D, E, K are known as the water fat soluble, right? A, D, E, K are the fat soluble. Then vitamin B complex, right? Vitamin B complex and the vitamin C, right? Vitamin C are the water soluble vitamins. So because of their water solubility, it can be easily excreted through the urine. So we cannot store the particular vitamin B complex and C in our body except vitamin B12. There is an exception. Vitamin B12, right? We have already seen, right? Vitamin B12 can be stored in our body even though it is a water soluble vitamin. Right? Then riboflavin, it is vitamin B2, right? It will be vitamin B2. So here option C will be the correct statement given the following. Here is the next question. Based on the protein structure, which structure is absolutely necessary for the biological activity? Biological activities of the proteins. Tertiary structure, right? Tertiary structure is responsible for the biological activity of the protein. So if there are any change in the tertiary structure, it leads to the deformation or the destruction of the particular protein, right? Okay, that is the same happening in the denaturation of protein. During denaturation of protein, if you are changing the pH or temperature, there will be the changes in the tertiary structure of protein. So it results in the losing biological activity of the protein. Okay. Here is the question based on the nucleic acid topic. Different images are given here, diagrammatic representations are given here. You have to identify which one is the diagrammatic representation of a nucleotide. Nucleotide. So in the case of a nucleotide, there will be the nitrogen base, sugar as well as phosphoric acid, right? So while observing these structure, you can see that the only one option, that is option D, is having the phosphate group here. This is our sugar. It may be ribose or deoxyribose. If it is the RNA, it will be ribose sugar. If it is DNA, it will be deoxyribose sugar. And this is the nitrogen base, right? It will be adenine, so we can identify that it will be the deoxyribose nucleic acid. So option D will be the correct answer here for the nucleotide. Here are some of the questions which is based on the fill in the blanks question. Here is the first question. The simplest and least carbon containing amino acid is? So glycine is the simplest carbon containing amino acid having one COOH group and one NH2 group. Then the next question, the amino acid molecules which possess positive and negative charges at their amino and carboxyl groups respectively are said to be in which form? This is the major property of the amino acids known as the Switter ion property, right? Switter ion property. Okay, this is also a very important part of the amino acid group, Switter ionic form. So there you must have an idea about the isoelectric point during the pH change and how these ions are forming, right? The next question, which of the biomolecule that is having the hierarchy of primary, secondary, tertiary and quaternary structures? Proteins, right? Proteins are the biomolecules having different structures like primary, secondary, tertiary and quaternary structure. The last question, the sugar which is present in the milk is? We have studied about the different types of polysaccharides and disaccharides. Which one is the sugar that is present in the milk? It is also known as milk sugar. Lactose, right? Lactose is the sugar which is present, or the carbohydrate which is present in the milk. Okay, here is the last question. The difference between amylose and amylopectin is? These amylose and amylopectin are the two parts, right? Water soluble and water insoluble part in the case of starch molecule. What are the major difference between the amylose and amylopectin? So here you can see this is the structure of amylopectin. You can see that there is a 1,4-glycosidic linkage, right? There is a 1,4-glycosidic linkage in between the alpha D glucose, and again 1,6 alpha linkage is in between the alpha D glucose, which means that this is the amylopectin is the branched chain containing 1,4 alpha linkage and 1,6 alpha linkage. So option A will be the correct answer here. Clear? Okay. That was the last question from this session. So in the case of biomolecules, this is the one important topic you have to keep in mind for your examination point of view. That is about the carbohydrates and the glycosidic linkage. So just go through the particular session and go through the structure of this particular carbohydrates. Here it is amylose having the 1,4-glycosidic linkage in between the 2 alpha D glucose. In sucrose, it will be 1,2-glycosidic linkage between alpha D glucose and beta D fructose. You must have idea about the structure, right? What is the difference between alpha and beta forms? In maltose, it will be the 1,4-glycosidic linkage between alpha D glucose. You can connect with this, right? These are same. Almost same in case of linkages. Then in lactose, it will be beta D glucose and beta D galactose, 1,4-glycosidic linkage. Again in cellulose, it is 1,4-glycosidic linkage is formed between beta D glucose, 2 beta D glucose molecules. Clear? So far we have successfully completed our biomolecules chapter. We have studied about the carbohydrates in detail. So this is the one of the important topic you need to keep in mind, you need to memorize some terms, right? Some terms to get maximum marks in the particular chapter. So carbohydrates, you need to identify the particular glycosidic linkages and their structure. In case of proteins and amino acids, you need to memorize the names of amino acids in terms of their structure also. Then proteins, structure of proteins, primary, secondary, tertiary and quaternary, what are the differences? Then the last one, it is about the enzymes and vitamins, right? Enzymes also you need to memorize. Then vitamins also, along with the chemical names and their deficiency disease, it is very important. The last topic, it is about the nucleic acids. What are the different types of nucleotides? What are the nitrogen bases? What is the bond which is formed between the nucleotides, right? For example, if you are taking the DNA or RNA, what are the different bonds formed between the DNA and RNA? Clear? Okay, I hope it was clear for everyone. So, again the reminder about the AATS. You will get the 50% off for using the YouTube 50 off code. So, link will be provided in the description. If anyone missed this particular AATS series, just enroll fast. Okay, that's all about from my side for today. Okay, I hope it was clear for everyone. If you really like the session, hit the like button, share this particular session to your friends also, then subscribe our YouTube channel. So, in the next session, I mean tomorrow, I will be coming back with the new chapter in the grade 11, that is hydrocarbons. This is a very important topic in the organic chemistry topic, right? So, if you have any doubt, you can ask, otherwise we can wind up the session for today. Okay, anyway, bye-bye all of you. Take care. See you in the next class. Thank you so much.